Okay, so hi everyone. My name is Leilani, and I am a rising senior studying cell molecular biology and data science. So for my project this summer, I decided to look at feature representation in protein models. Okay, so just beginning with defining the initial problem. In the past few years, countless large language models trained for different protein predictive tasks have been developed and made publicly available. So ranging from AlphaFold's revolutionary structure prediction feature to ProtGPT's ability to generate novel protein sequences, these tools have greatly accelerated scientific research. But the influx of these models now poses a very complex problem. Just how do researchers decide which tool to use? So I decided to address this issue by creating my own tool that explores the model's capabilities. So by understanding exactly what these models are learning about proteins, I believe that we can streamline the next generation of scientific research by making these very complicated tools more accessible and digestible for users of all skill ranges. So I decided to begin by exploring three different very well-known models, and those are ESM2, ProtBERT, and ProtGPT2. So the input to all of these models are the same, which is an amino acid sequence, and those are the primary structures of proteins. So they're basically just a list of letters that represent different amino acids with different biochemical properties. So in this case, large language models that are usually used for human language can now be applied to the language of biology. So ESM2 and ProtBERT are encoder models that basically just process the whole sequence context, while ProtGPT2 is a decoder model, which is trained to predict the next amino acid, very much like text generation. So all the models output the same thing, which are embedding vectors, which essentially just are numerical representations that represent each residue, or another word for amino acid. So in this basic pipeline that I created here, you can just see an example of an embedding space that captures all of these vectors. And just for context, similar amino acids will be closer to each other in this space, and different amino acids will be further apart. So all models have a transformer architecture and use methods of unsupervised learning on unlabeled data, which in this case are the amino acid sequences. So this is common architecture for large language models because the first step in any prediction task is to encode and transform the input into a machine readable form from which these models can learn. So for the primary objective for ESM2 and ProtBERT are the same, which are to capture the structural and functional features of the proteins based off of their amino acid sequences. And ProtGPT2 is a little different. It's mainly used for protein generation and design. So all of the models were trained on similar publicly available protein databases. And the only difference is here that ProtGPT2 is also trained on synthetic sequences. And this is just because it has a very specialized task of de novo protein sequence generation, and it tries to follow the principles of natural protein. So it has to add additional training data in there. So the tokenization step is also a key part of these large language models. And the encoder models differ from the decoder model in this step. So the tokenization process essentially just breaks down the initial sequence into smaller fundamental units, which can then be processed. So ESM2 and ProtBERT both process an amino acid sequence one residue at a time, while ProtGPT2 processes the sequence in bytes. And I tried to look into this, and it's kind of just decided to black box it because it's a very complicated subword tokenization algorithm. But that did affect my results later on, and I'll get into that. But essentially, the outputs are all the same. So they output an embedding per residue, which again are just these numerical representations of the amino acids in the embedding space. And the values in these vectors represent exactly what the model has learned about the protein based solely on its sequence. So I decided to first look at these embeddings and just visualize them. So I compared the per residue embeddings of the two models, ESM2 and ProtBERT, to determine exactly the similarities in their feature representation. So I arbitrarily decided on lysozyme as my test case amino or sorry, for amino acid sequence, just because it's a very well documented protein whose features and structure we know really well. And so I decided to use the models to create embeddings for lysozyme based off of its amino acid and compare the results. So after looking at how similar the residue level embeddings were between these two models and reducing them to the same dimension, which is shown here on this graph, I calculated the average cosine similarity. So average or cosine similarity is just a metric used to calculate the similarity between two vectors in space. And the values range from 0 to 1, 0 being low and 1 being very high similarity. So in this case, the average cosine similarity was just around zero point 0.1, which is pretty low. And this is interesting because this means that the two models are probably capturing very different kinds of features based off the same protein, the same amino acid. So their internal representations don't align much for this protein, which is a good sign that they're complementary, but not the same. And then I performed k-means clustering. And as you can see here as well, the different clustering patterns suggest very different feature representations between models with for example, the ESM2 clusters. There are kind of three major clusters that you can make out. Whereas in the prop-BERT embeddings, it's kind of a little more complicated. So yeah, this kind of just led me to believe that these models are capturing very different things about the same amino acid sequence, which they then use for downstream tasks such as prediction of structure, function, interaction. Yeah, so then one of the major limitations, which I mentioned earlier about ProtGPT-2, is that in order to perform k-means and compute similarity, the embeddings must have the same shape. So that means that I could only compare ESM2 and ProtBERT due to the special tokenization process that the other model uses. However, based on these results, I do believe that, well, like one can hypothesize that ProtGPT-2 also captures different features. So in the future, I'd like to add a handle case for those models that output embeddings of different shapes. So this, of course, led me to my hypothesis, which is that the reason for these differences in each model varies in the specific biochemical properties that are encoded in their embeddings. So to explore this question, I looked at four common biochemical properties of amino acids that are all involved in determining a protein's secondary and tertiary structure, as well as many other functions in the body. And these are hydrophobicity, polarity, charge, and molecular weight. So once again, just a reminder from a few slides ago that these were the results of the clustering, the k-means clustering that I performed on the embeddings. And it very much shows that these models are capturing different features of the sequence. So then I decided to perform a series of ANOVA tests for the two different models and all the different features that I selected. And I found that these models do in fact vary in what features they are most capable of preserving. So for example, here you can see that ESM2 performed relatively consistent across the four categories, with its most prominent feature captured being charge and its least being molecular weight. ProtBERT performed less consistently and showed that its embeddings were more significantly correlated with hydrophobicity and less so with polarity and charge. So this was kind of interesting to see. I also decided to look at active site similarity. So active site in a protein is a specific region on an enzyme where a substrate binds and a chemical reaction occurs. So specifically in lysozyme, the two catalytic residues are gluv35 and asp52. So my hypothesis was that these amino acids would be represented similarly in the embedding space due to their special function. And their similarity will indicate the model's ability to detect these functionally important residues. So I found that results demonstrated that ESM2 much better captured the active site residues with a higher average cosine similarity score of 0.831. The similarity of the residues in the other model, ProtBERT, was lower, but still shows some ability to detect their similar function because the value was 0.6537. So it wasn't low, but it definitely wasn't as high as ESM2. And I was also really interested in mutant comparison, and this kind of came on later in the project, so still a work in progress, but I wanted to see if the models were able to understand the mutation. So I decided to just manually substitute one of the active site residues, sort of mimicking a substitution mutation in an amino acid sequence. And the similarity scores between each residue embedding in the mutant versus wild type sequence was found to be higher on average in the prop BERT model. So but both values were pretty high with zero point 0.8 and 0.97. And this was interesting because I found that it suggests that the models are not really that sensitive to mutations. And this might prove to be um, a big limitation in their downstream predictive tasks because a lot of current research is being done about the effect of mutations on structure and function. And if these models aren't able to capture that mutation and the significance of it, they might not perform as well in their downstream tasks. So right now, my current focus is on further analyzing these results, especially with statistical analysis. So after thoroughly exploring the model's features and probably following one too many tangents with a few things, I decided to package these analytical tools into a Python package. So the features of this package essentially just comprise of the steps that I myself took to determine the difference between these protein prediction models. So some of the outputs of the package are shown here. So figure one is just the 3D visualization colored by residue features. And it's based off the results of the ANOVA test because it selects the feature with the most significant or selects the feature with the most significance to visualize. So that is just an example of what the package outputs. And then as well in figure two, that's just a heat map of the residue similarity. And this is based off of the cosine similarity that I calculated between the residues. And then there's also a highlighted part of that heat map for the key residues, such as active site residues in this case. And then the last thing that it outputs is a um, just an embedding comparison for the mutant versus wild type sequences. So just something else that users can use to understand how exactly their model is capturing the sequence and understanding their protein. So there's a few future directions that I definitely want to continue exploring after this. So definitely want to refine the mutant comparison because this is, I think, going to play a key role in a lot of the downstream tasks, such as structure prediction for AlphaFold, things such as that. I also want to try to create a user interface. I'm not really too familiar with how to do that, but I do want to try to, I think that the user application interaction is going to be really important for this. And then I also want to expand beyond lysozyme and perform embedding analysis between many different proteins, especially proteins that play very different roles in the body and have different locations. I think that'd be interesting to see how well the models understand the other proteins as well. And then along similar lines, I want to add more open source protein models to increase options for users because there are so many that continue to be developed even today. So it's just keeping up with the current models is definitely really important as well. Okay, and then these are my acknowledgments. So I would like to thank everyone that helped me with this. I definitely learned a great deal. And these were my sources, and that's it. Any questions?
So really nice presentation. For user interface, what were you kind of envisioning for the interactions for users with the package that you developed?
Oh, I'm not too sure yet. I'm thinking something along the lines where a user can input some sort of amino acid sequence, and I want to have a feature where you can add a mutation to it, and it finds some sort of database of different sequences and most closely match it to the one that you input. So maybe some sort of website, or I'm not too sure yet, but yeah, something where it's just kind of easy for users to visualize how exactly these models are understanding the protein and what model is best for their specific project.
Okay. Are you publishing Python package to pip or some package manager?
Yes, yes, it's
Okay,
all on GitHub right now. So
nice.
yeah, yeah.
Okay, thank you.
Awesome job, Leilani.